My name is Diana O'Dowd, and I'm a professor in the Department of Developmental and Cell Biology and Anatomy and Neurobiology at UC Irvine. And today I'm going to demonstrate the preparation of primary neuronal cultures from Drosophila embryos. And what this entails is actually removing all of the cells from a Midgastrula stage Drosophila embryo and placing these in cell culture. And what we use these cultures for are to really understand what are the genes and environmental factors that are important for regulation of electrical excitability and synaptic transmission between Drosophila neurons. To start this procedure, we have to collect Drosophila embryos. To do that, we take egg collection plates, spread a little bit of yeast paste on them. It's a favorable substrate for the adult flies to lay their eggs. We put these plates on bottles containing a population of adult flies, usually 100 to 200 flies, and then we let the adult flies lay their eggs for about 2 to 3 hours. Then we remove the egg collection plates, and these are the embryos that we use for the culture procedure. The next step in the procedure is to dechorionate the embryos, and that means we actually remove the chorion. We do this by washing the embryos off into a Buchner funnel on which we have a piece of filter paper so it catches the embryos. We allow them to sit in the Buchner funnel in a 50%